Hi, everyone. I'm here to briefly discuss something that's very important to or integral to my research, and it has become a pretty prominent topic of research in the world of microbiology and neurodevelopment and maternal influence on both. And so I'm here to discuss how maternal diet and nutrition has a subsequent influence on the gut microbiome and the likely association to neurodevelopment and cognitive development. So I believe it is important to discuss the sterile womb paradigm, which is this concept that was the widely accepted dogma that the human fetal environment is sterile and that the neonate's microbiome is sterile and it is acquired during birth. And this was the widely accepted notion up until recent studies, which challenged this sterile womb paradigm. And these studies have found that in healthy pregnancies, there is identification of bacterial communities and populations found within the placenta, amniotic fluid, and the meconium. These findings have led many scientists and researchers to challenge this paradigm. This proposes that the microbiome acquisition begins in utero. And this could fundamentally change our understanding of gut microbiota acquisition and its role in human development. So here you can kind of see the sterile womb paradigm schematic representation. Basically here in the sterile womb paradigm, which was the more widely accepted dogma up until recent studies, the placenta, amniotic fluid, and fetal gut remain sterile during a healthy pregnancy, and the early microbiome of the offspring is acquired at birth. However, in this now more interesting and widely accepted with research idea is the in utero colonization hypothesis. And this proposes that some microbial members of the infant's gut microbiome are acquired before birth in utero. And probably this is due to direct contact with the placental microbiome, which is originating from the mother's gut microbiome. So here, this is just another schematic showing how maternal nutrition can shape the neonatal microbiome. Diet has an effect on the maternal uterus environment, and there are microbes that are, like I said, present in the placenta, amniotic fluid, and fetal membrane. And this window of shaping the neonatal microbiome and neurodevelopment actually mirror each other. And so that is why there's this proposal that there's likely a link between the two. There has been shown to be differences in the microbiomes of those born via cesarean delivery versus those born vaginal delivery. And this opportunity for microbial composition modulation continues postnatally, and with the chief influence being the method in which the baby is fed, whether that be through breastfeeding or through formula feeding. Ultimately, the resulting microbial composition can be indicative of potential disease development in offspring. So this is just one study that I think is pretty prominent in explaining that there's likely a link between the infant gut microbiome and how it is indicative of potential cognitive development. So infants of 1 year of age, this is using human infants, by the way, as subjects, they were testing for a link between microbial composition at 1 year of age and associated cognitive outcomes using a standardized scale of measuring learning. Fecal samples were collected and analyzed using 16 sRNA sequencing in order to identify and quantify the bacterial taxa, and also put cluster analysis to identify 3 distinct groups of infants defined by their bacterial composition. So cluster 1, characterized by relatively high abundance of Faecalibacterium, cluster 2, characterized by relatively high abundance of Bacteroides bacterium, and cluster 3, characterized by relatively high abundance of an unnamed genus and family, Ruminococci bacterium. And so the microbial composition and clusters at 1 year of age actually were predictive of cognitive performance at 2 years of age. So using the Mullen scale, which is basically like an analysis scale for learning, the Mullen early learning composite by cluster here, you can see that cluster 2, which had the highest level of Bacteroides, performed significantly higher in overall cognitive evaluation. The examination of specific functional groups in this lower graph using the Mullen scale also showed that cluster 2 demonstrated significantly higher scores in both receptive language and expressive language, as well as the highest score in visual reception as compared to the other 2 clusters that were characterized by other bacteria. And then this is another study that is furthering evidence involved with maternal influence on the infant gut microbiome. This one is pretty interesting. This is involving prenatal stress. And it showed that there was an altered microbial composition in the neonate gut, and it corresponded with changes in metabolic profiles. So this was using a mouse model of early prenatal stress to examine the outcomes both on the mother and the offspring. The researchers hypothesized that the maternal stress would in fact alter the vaginal microbiota composition, and in addition, a vertical transmission of vaginal bacteria likely disrupted dysbiotic community, may propose distinct bacterial colonization patterns in the offspring gut. So in this first graph, you can see that there is a significant difference in the vaginal lactobacillus, the most prominent taxa, which is the most prominent taxa in the vaginal environment, between those mice who were and were not exposed to the early prenatal stress, with a significantly lower expression in the stress group. This second graph here shows the expression of lactobacillus in the offspring between treatment groups as well as between sex. And it also shows that these variations were observed regardless of gender. And then in this third graph, it's showing that there is a significant positive and close correlation between offspring and maternal lactobacillus expression for each treatment group. The researchers also observed that the altered microbiota composition in the neonate gut corresponded with modulations in metabolite profiles that are involved in energy balance, and with region-specific disruptions of amino acid profiles that are correlated with developing brain regions, which leads them to propose that the vaginal microbiota may be a novel factor by which maternal stress can contribute to reprogramming of neurodevelopment in the offspring. So there are several studies. These are just 2, one showing the effect of direct maternal stress and one showing how the infant's gut microbiome is possibly indicative of neurodevelopment in that infant. Again, I could go on for quite a while, but I hope, if nothing else, you all found this interesting. If anyone has any questions, you can email me. I look forward to hearing everyone's feedback and as well as watching everyone else's presentations. It's been so interesting to see and hear everyone's presentations, or at least the last time, because everyone else's work and majors and research are so different than mine. So it's very interesting. But yeah, thanks everyone.